dark reaction or Calvin cycle is the carbon fixation pathway, CO2 fixation pathway. It is also known as reductive pentose phosphate pathway, the C3 pathway, because the first formed stable compound is a 3-carbon compound. Photosynthetic carbon reduction cycle, because the reduction of carbon takes place during this reaction, and also by Calvin cycle, because it is discovered by Melvin Calvin. So for the dark reaction, carbon dioxide is required, and carbon dioxide enters into the leaf through the stomata during gaseous exchange. So the first step in the reaction is the incorporation of carbon dioxide into a 5-carbon compound known as ribulose biphosphate. This reaction is catalyzed by the enzyme ribulose 1,5-diphosphate carboxylase bar oxygenase, or Rubisco. Rubisco is the most widely distributed protein in plants, because more than 50% of protein present in plants is Rubisco. It is the most important protein or enzyme for a photosynthetic organism. It can carboxylate as well as oxygenate the Calvin cycle intermediates, especially the ribulose biphosphate. We will see both the reactions. Now we look into the carboxylation reaction, and in the photorespiration we will see how the oxidation of ribulose biphosphate takes place with the help of Rubisco. So the Rubisco combine ribulose 1,5-diphosphate with carbon dioxide to form 2 3-carbon compounds, because in ribulose 1,5-diphosphate there are 5 carbon atoms. 1 carbon comes, it becomes 6 carbons, and the 6-carbon compound is an intermediate compound which is highly unstable, which is not yet isolated or identified. It splits into 2 3-carbon compounds, phosphoglyceric acid or 3-phosphoglycerate. 3-phosphoglycerate is the first stable compound in Calvin cycle, so this is known as C3 cycle. The Rubisco cycles between an inactive state and an active state. Rubisco is active only when light is present. The activation of Rubisco requires the presence of magnesium ions, carbon dioxide, and the correct stromal pH. At night, 2-carboxy-arabinitol 1-phosphate inhibits Rubisco. So with the help of Rubisco, 2 molecules of phosphoglycerate, 3-phosphoglycerate is formed, and the next step is the synthesis of hexose sugar from this phosphoglycerate. For that, more energy is required, so this 3-phosphoglycerate undergo phosphorylation reaction and becomes 1,3-biphosphoglycerate, utilizing 1 molecule of ATP. After that, again it undergo one more reaction to make it energy rich, undergo reduction, and the glycerate is converted into aldehyde utilizing one NADPH2 molecule and becomes 3-phosphoglyceraldehyde. So to remember the process, we can logically think like this. First reaction is the carboxylation of ribulose 1,5-diphosphate. This ribulose 1,5-diphosphate is a 5-carbon compound, so 1 more carbon comes in the form of carbon dioxide. It combines and forms a 6-carbon unstable compound, but splits into 2 molecules of 3-phosphoglyceric acid, which is not an energy rich compound. For the formation of hexose sugar, more energy is required, so it undergoes 2 steps of energy addition. Like we can think like that. 2 steps of energy addition. The phosphoglyceric acid first undergo phosphorylation and it becomes 1,3-diphosphoglyceric acid. 1 ATP is utilized, so 1 energy rich bond is formed there. 1 phosphate comes. This 1,3-diphosphoglycerate undergo 1 more reaction to make it more energy rich. It undergoes a reduction reaction where the glycerate, the acid, is converted into an aldehyde, a 3-phosphoglyceraldehyde. So this is a reduction, so hydrogen is required. Hydrogen comes from NADPH2, so NADPH2 is utilized. So we can logically remember the process. Now it becomes 3-phosphoglyceraldehyde. And there is an isomer of the 3-phosphoglyceraldehyde, which is a keto form known as the dihydroxyacetone phosphate or DHAP. So phosphoglyceraldehyde and DHAP present in equilibrium in the system. Then these two molecules, this keto form of 3-carbon compound, 3-phosphoglyceraldehyde and DHAP combined together to form the first sugar molecule, fructose 1,6-biphosphate. The fructose 1,6-biphosphate releases one phosphate group and becomes fructose 6-phosphate. The fructose 6-phosphate is converted into glucose 6-phosphate and further into glucose 1-phosphate. All these 6-carbon sugars are part of the hexose monophosphate pool of the cell. Now we can think about the enzymes involved. The 3-phosphoglycerate is converted into 1,3-biphosphoglyceric acid utilizing 1 ATP molecules with the help of the enzyme 3-phosphoglycerate kinase. Similarly, the 1,3-biphosphoglycerate is converted into 3-phosphoglyceraldehyde by the utilization of an NADPH2 molecule with the help of the enzyme 1,3-biphosphoglycerate dehydrogenase. So these are the two enzymes, first two enzymes involved in the conversion of PGA into PGAL. We have considered only one molecule of ribulose 1,5-diphosphate. We think about a glucose molecule, there are 6 carbons. Just think that the reaction continues and a pool of all these compounds such as DHAP, PGAL and fructose 1,6-biphosphate are formed by the continuous carboxylation of ribulose 1,5-diphosphate. So we have the dihydroxyacetone phosphate, 3-PGAL and fructose 6-phosphate. Among these, fructose 6-phosphate is a ketone. The dihydroxyacetone phosphate also has a keto group and PGAL is an aldehyde. So the synthesis of hexose sugar is completed. Next is the regeneration of the ribulose 1,5-diphosphate because we have used one ribulose 1,5-biphosphate. To repeat the cycle, we have to regenerate the ribulose 1,5-biphosphate. So from these compounds, we have the phosphoglyceraldehyde, dihydroxyacetone phosphate and fructose 6-phosphate. We have to regenerate ribulose 1,5-biphosphate. To think logically about the coming reactions, remember this particular character. The fructose 6-phosphate is a ketone, dihydroxyacetone phosphate is also a ketone and PGAL is an aldehyde. In the next reaction, the fructose 6-phosphate reacts. It can react only with an aldehyde, so it combines with PGAL. So when it reacts, fructose 6-phosphate has 6 carbon atoms and phosphoglyceraldehyde has 3 carbon atoms, total 9 carbon atoms. But with the help of the enzyme transketolase, this 6-carbon compound fructose 6-phosphate and 3-carbon compound phosphoglyceraldehyde combine together and splits into a 5-carbon compound and a 4-carbon compound. The 5-carbon compound is cellulose 5-phosphate and the 4-carbon compound is erythrose 4-phosphate. This reaction is catalyzed by the enzyme transketolase. Remember the compound we have to reach is a 5-carbon compound. It is ribulose 1,5-diphosphate. Here we got one 5-carbon compound, cellulose 5-phosphate. The remaining compound, the other compound is erythrose 4-phosphate. These two compounds, one is a ketone and other is an aldehyde. Erythrose 4-phosphate is the aldehyde and cellulose 5-phosphate is the ketone. So the erythrose 4-phosphate, which is an aldehyde, can react only with the ketone. The 3-carbon ketone is the dihydroxyacetone phosphate. So erythrose 4-phosphate react with dihydroxyacetone phosphate. If you think like this, you will not confuse with which compound this erythrose 4-phosphate reacts. Erythrose 4-phosphate is an aldehyde, so it can react with the ketone. The ketone we have is dihydroxyacetone phosphate. So erythrose 4-phosphate and dihydroxyacetone phosphate combine together with the help of enzyme aldolase to form pseudoheptulose 1,7-diphosphate. Pseudoheptulose is a 7-carbon compound, and it is clear that erythrose 4-phosphate is a 4-carbon compound and dihydroxyacetone phosphate is a 3-carbon compound. 4 plus 3, 7 carbon compound is formed, which is pseudoheptulose 1,7-biphosphate. The pseudoheptulose 1,7-biphosphate releases one phosphate group and becomes pseudoheptulose 7-phosphate, which is a 7-carbon compound. With the help of the enzyme pseudoheptulose 1,7-biphosphate phosphatase enzyme, now we have pseudoheptulose 7-phosphate. The pseudoheptulose 7-phosphate is a ketone. So it has to react with the aldehyde. So it reacts with phosphoglyceraldehyde. We have 7 carbons in the pseudoheptulose 7-phosphate, 3 carbons in the phosphoglyceraldehyde. Total there are 10 carbons, and these 10 carbons form 2 compounds such as cellulose 5-phosphate and ribose 5-phosphate. So the enzyme must be transketolase. The pseudoheptulose 7-phosphate reacts with phosphoglyceraldehyde in the presence of the enzyme transketolase and form one molecule of cellulose 5-phosphate and one molecule of ribose 5-phosphate. So we have 3 molecules, 3 5-carbon molecules. The cellulose 5-phosphate formed from fructose 6-phosphate and glyceraldehyde 3-phosphate, and now one more cellulose 5-phosphate is formed. And we have another ribose 5-phosphate is also here. The cellulose 5-phosphates are converted into ribulose 5-phosphate with the help of the enzyme pentose phosphate epimerase, whereas the ribose 5-phosphate is converted into ribulose 5-phosphate by the enzyme pentose phosphate isomerase. So total 3 molecules of ribulose 5-phosphate are formed. This ribulose 5-phosphate is converted into ribulose 1,5-biphosphate with the help of the enzyme phosphoribulose kinase. Since one molecule of glucose is formed as a new compound, product of reaction, we can think about 6 carbon dioxide molecules at a time. So 6 carbon dioxide molecules combine with the 6 molecules of ribulose 1,5-biphosphate, and further reactions takes place. 6 molecules of ribulose 1,5-biphosphates are utilized, 6 molecules of carbon dioxide is fixed, and 1 molecule of glucose is synthesized. So 6 molecules of ribulose 1,5-phosphate, 6 into 5, 30 carbon atoms. 6 molecules of carbon dioxide, again 6 carbon atoms. 30 plus 6, 36 carbon atoms. 1 hexose is formed, so 30 is remaining, and these 30 carbons are utilized for the regeneration of the 6 ribulose 1,5-biphosphate molecules. Here we can see a diagrammatic representation of the cycle. Here it is illustrated with 3 molecules of ribulose 1,5-biphosphate combining with 3 molecules of carbon dioxide, which is minimum for indicating the presence of ribulose, cellulose and ribose. So it is a simplified form, but when we think about the cycle with respect to 1 molecule of glucose or 1 molecule of fructose, we should consider 6 carbon dioxide molecules combining with the 6 molecules of ribulose 1,5-biphosphate. So I am explaining it once more for your easy understanding. We can classify the Calvin cycle into three stages for our convenience, such as the fixation of carbon dioxide, then the reduction of this fixed carbon, then the formation which result in the synthesis of sugars and the regeneration of RUBP. So the first step we can see here, the ribulose 5-phosphate. For our convenience, we take 3 molecules of ribulose 5-phosphate at a time. This ribulose 5-phosphate molecules is converted into ribulose 1,5-diphosphate with 3 ATP molecules. This ribulose 1,5-diphosphate is the first molecule with which the carbon dioxide reacts. So 3 molecules of ribulose 1,5-biphosphates are there. 3 molecules of carbon dioxide combined with this ribulose biphosphate with the help of the enzyme Rubisco, ribulose biphosphate carboxylase bar oxygenase to form 3-phosphoglycerate molecule. So since ribulose 1,5-biphosphate is a 5-carbon compound is added with the carbon dioxide, so 6 molecules of 3-phosphoglycerate are formed. 3-phosphoglycerate, 3-carbon compound into 6 18-carbon molecules. This 3-phosphoglycerate molecule combined with ATP molecules to form 1,3-biphosphoglycerate. Then this 1,3-biphosphoglycerate combined with 6 NADPH molecules to form phosphoglyceraldehyde phosphate, 3-phosphoglyceraldehyde phosphate. This 3-phosphoglyceraldehyde phosphate is converted into DHAP and glyceraldehyde phosphate, dihydroxyacetone phosphate and 3-phosphoglyceraldehyde phosphate. One molecule each of 3-glyceraldehyde phosphate and DHAP combine together to form fructose 1,6-diphosphate. This fructose 1,6-biphosphate is converted into fructose 6-phosphate by releasing one phosphate group. Then this fructose 6-phosphate is a ketone, so it reacts with the glyceraldehyde phosphate. So it combined with one molecule of glyceraldehyde phosphate with the help of the transketolase enzyme. Here we have 6 carbons. Here we have 3 carbons. So 6 plus 3, 9 carbons. It combine in the presence of transketolase enzyme and form xylose 5-phosphate and erythrose 4-phosphate. Xylose 5-phosphate have 5 carbons and erythrose 4-phosphate have 3 carbons. The xylose 5-phosphate directly converted into ribulose 5-phosphate with the help of phosphopentose epimerase. The erythrose 4-phosphate, which is an aldehyde, combined with the DHAP and we have 4 carbons in erythrose and 3 carbons in DHAP, so total 7 carbons. It form pseudoheptose 1,7-biphosphate. It releases one phosphate group and it becomes pseudoheptose 7-phosphate. It has 7 carbons and it is a ketone. So it combines again with the 3-carbon compound glyceraldehyde phosphate in the presence of the enzyme transketolase to form 2 5-carbon compounds. Here pseudoheptose has 7 carbon. Glyceraldehyde phosphate has 3 carbon, so 7 plus 3, 10. This 10 carbons split into 2 5-carbon compounds, ribose 5-phosphate and xylulose 5-phosphate. Both these, xylose 5-phosphate is converted into ribulose 5-phosphate with the help of the enzyme pentose phosphate epimerase, and ribose 5-phosphate is converted into ribulose 5-phosphate with the help of enzyme pentose phosphate isomerase. So the ribulose 5-phosphate is formed. This ribulose 5-phosphate combined with the ATP molecules to form ribulose 1,5-biphosphate. So the reaction, the cycle can be continued. This is the Calvin cycle. We will think about the energetics, such as how many ATP molecules are synthesized during light reaction, how many are synthesized in cyclic photophosphorylation, how many are synthesized in the non-cyclic photophosphorylation, how many are utilized during the dark reaction, etc. in the next class. Thank you.